this screencast, we will talk about how a muscle contracts, specifically the sliding filament theory or the sliding filament model. So this process can be a bit complicated or can seem a bit complicated, but it really is able to just be broken down into steps. I think it's very important if you think about the end goal and what needs to happen in the end, and then which obstacles need to be overcome in order for that to happen. That will help make the sequence a lot easier. You'll also see in this screencast and in other videos that there are a wide variety of number of steps. The number of steps is not as important as the sequence itself. So it doesn't really matter how many steps you break it down to. Break it into however many steps you need to understand it. But this screencast will use 10 steps because that's how I broke it down. So first of all, an easy overview of muscle contraction, or the overall big picture, is that myosin is going to bind to actin in order to pull on the actin. This is going to cause actin to slide past the myosin. The overlap between the two fibers or types of fibers in the sarcomere will increase, which causes the muscle fibers to shorten as each individual sarcomere shortens, and the muscles will pull on their attachments on the bone in order to move body parts. Some of the big players we're going to see in this are myosin, actin, troponin, and tropomyosin. All four of those are proteins that are making up the thick and the thin filaments within the muscle. The sarcomere, which is the functional unit of contraction, calcium, ATP, and acetylcholine, or ACH, which is our neurotransmitter. Myosin fibers are thick, twisted fibers, and their heads project outwards. So as you can see here, we have myosin in this picture, and they have these little heads that stick out. These heads are going to bind to the actin in order to pull it. The actin is twisted into a double helix. It's going to contain two other proteins, troponin and tropomyosin. As you can see here, the actin are these like pearl kind of looking strands. The tropomyosin is this purplish rope, and the troponin are those blue little blobs. The actin fibers have a binding site on them for the myosin heads. You can't really see the binding sites in this diagram because right now the tropomyosin is covering it. That's going to become important. We're going to be talking about a sequence of events that ultimately leads to the actin binding sites being uncovered so that myosin can connect to them. This whole process we're going to talk about is called the sliding filament theory. Just a reminder that in science, when we're talking about theories, we're talking about things that have been tested and proven by experiments and lots of different evidences over a long period of time. Myosin and actin filaments, as I already mentioned, are going to slide past one another to shorten the sarcomere. It's important to look at this diagram carefully and see that the actin is not shortening. The myosin is not shortening. It's just that the space between them is getting more overlapped. So you can see here, these fibers are still the same length, no matter how contracted the muscle is. It's just that the actin is sliding inwards to shorten the muscle as a whole. So now let's talk about the steps. Again, I numbered these steps just to make it a little easier, but there are a wide variety of numbers of steps. The sequence or the order is what is important. The first thing that's going to happen is a nerve impulse is going to travel from the brain or the spinal cord all the way down all of your motor neurons until it reaches the very last motor neuron. When this nerve impulse reaches the last motor neuron, it's going to go down the axon, down to the axon terminals, and reach the neuromuscular junction, which is a synapse between the neuron and the muscle. In this neuromuscular junction, when the impulse reaches the end of the axon terminal, the neurotransmitter acetylcholine is going to be released from the vesicles. So here you have acetylcholine that was sitting in the vesicles. When the impulse reaches the end of the axon terminal, it's going to release the acetylcholine into the synaptic cleft. It's then going to cross the synaptic cleft and bind to the receptors that are on the muscle fiber. When this happens, the impulse is now on the muscle side of the synapse. The sarcolemma is stimulated. If you remember, the sarcolemma is the cell membrane of a muscle cell. And so the impulse travels over the surface of the sarcolemma and deep into the fiber because of the sarcoplasmic reticulum and the T-tubules. So if you look at this whole thing is one muscle fiber. In here, we have all these other long skinny tubes called myofibrils. That's where the sarcomeres are. And so this impulse is now going to cover and go within the muscle fiber. When this happens, it causes calcium channels to open. So calcium that was previously outside the cell can now go through the cell membrane or the sarcolemma. It can't pass through on its own, so it needs these channels to open. The electrical impulse that just came over the sarcolemma is what causes the calcium channels to open. When calcium ions diffuse into the sarcoplasm, they then bind with troponin molecules. As you may recall, when proteins bind with something, it causes the shape of the protein to change. So when troponin binds with calcium, its shape changes, which pulls the tropomyosin off the binding sites for actin. So you can now see that these binding sites are open. Here they were covered, so the myosin heads weren't able to bind to the actin until they were uncovered like they are now. So now myosin can reach up and attach to the actin filament, forming what's called a cross bridge. The actin is then going to be pulled towards the center of the sarcomere, so the Z lines will get closer and closer together towards the M line. This is known as muscle contraction. The muscle fiber shortens. Contraction is just shortening. So we said that actin is pulled, but how is actin pulled? When you look at these steps, I just want you to look at this for the sake of understanding that we need ATP and why we need ATP. So you can see here that ATP binding to the myosin head is what causes the myosin head to return to a resting or cocked position. So it's what pulls it off the actin. And then when the ATP is broken down, that's when it's going to reattach to the myosin. This will become important when we talk about the need for ATP in order to continue with muscle contraction. So, quick recap, in order for the muscle to contract, the impulse has to travel down all of the neurons to the last neuron. When it reaches the last neuron in the neuromuscular junction, it will cause the vesicles within the neuron to release acetylcholine into the synaptic cleft. This acetylcholine then binds to the receptors that are on the muscle fiber. This causes the impulse to spread over the sarcolemma and into the muscle fiber itself. The impulse being spread throughout causes calcium channels to open and lets calcium flood into the cell. When calcium is in the cell, it binds to troponin, changes the shape of the protein of troponin, which pulls tropomyosin away from the binding sites so that it's no longer blocking them. Now myosin can bind to the actin, which ends up pulling it to shorten the muscle fiber. This keeps happening as long as we have three things being met. As long as we still have an impulse or a stimulus, the muscle can still keep contracting. As long as we have enough calcium, the muscle can still contract. And as long as we have enough energy, the muscle can still contract. I like to think of this muscle contraction process as like dominoes. So if you have 10 dominoes all lined up, you need each domino to be there if you want to tip it over and have them all fall. If you take away one of the dominoes, the whole stack of dominoes or the whole line of dominoes can't fall anymore. So it's important that you have each of these dominoes in place. If you're missing the stimulus domino, you don't have the first domino. So you can't start this whole contraction. If you don't have calcium, you're missing one of the dominoes in the middle. So the contraction might go halfway, but then we don't have calcium to actually bind to the troponin. Same thing with energy, with ATP. If we don't have enough ATP, we can't get the myosin head to go back to its cocked position to then reach up and grab the actin again. So when we don't have these conditions being met, relaxation will occur. Usually this is just because we tell our muscle to relax. We stop sending the impulse to contract. Relaxation is essentially the opposite. Everything gets kind of cleaned up and put back where it goes. Acetylcholine gets broken down by acetylcholinease. This causes the impulse to stop, so it's not reaching the muscle anymore. Calcium ions are transported out of the sarcoplasm, and ATP breaks the cross bridges or pulls the head back. With the calcium out of the cell, troponin and tropomyosin can go back to blocking the binding sites that are on the actin. Now that the myosin and actin are no longer connected, the muscle can remain relaxed and be ready to be stimulated again. And that is a brief overview of muscle contraction. I would suggest watching this screencast again, finding different videos or animations, whether on this channel or other places on the internet, or reading through your textbook or other diagrams to help you understand the whole process more thoroughly.